watching my channel Learn to Know Cbse Biology. I am Domination and I am back with yet another new video of the series Exam Stamina 2020 with the name Discussion Video 2. Today I will be discussing about the DNA replication. It will include DNA replication forthcoming model and the semiconservative experiment. So, without any further ado, let me start. Number 1, experiment by Stahl and Messelson. What experiment did they do? They had taken E. coli and had grown them in a nutritive medium. You know, any type of microbes grow well in a nutritive medium. They took the medium of 15 NH4Cl. Since you know nitrogen are of the isotopes of nitrogen are of 14N and 15N. They took the 15N, 15 NH4Cl, 15 NH4Cl. So since 14N and 15N, the atomic weight of 14N is lower than 15N. So the weight is like this, 14N is lower than 15N. Thus these two weights can be easily separated by centrifugating in a cesium chloride centrifugator. With the medium cesium fluoride, if you can centrifugate them, the difference in the weight due to 14N and 15N gets separated. Okay, so what did they do? They had grown E. coli in a 15 NH4Cl medium. Next, and had kept for several hours. Next, they transferred this E. coli from 15 NH4Cl then to a medium of 14 NH4Cl. So they took initially a 15 NH4Cl medium, this E. coli they had grown, transferred this E. coli to a 14 NH4Cl medium. And there is a basic information that E. coli takes 20 minutes to form a generation. Okay, E. coli always takes 20 minutes to form a generation. When this E. coli was given in 14 NH4Cl and was taken for 20 minutes and was left there for 20 minutes so that they can grow, it was then centrifugated in a 14 NH4 in a cesium chloride centrifugator. So the E. coli with the DNAs of 14 NH4Cl, what does DNA contain? Nitrogen. Okay, the 14 NH4Cl and 15 NH4Cl, they were separated. They found that an intermediate hybrid of both are present. At last I will be drawing diagram to explain. The intermediate hybrid is present. Next they again gave a 20 minutes in 14 NH4Cl and checked. They checked that they found that after centrifugating again, they found that equal amount and equal number of the heavy DNA or 15 NH4Cl DNA and light DNA, that is 14 NH4Cl DNA, was found. Now, what does it signifies? It shows that initially when the DNA, suppose I use the black color to represent 15 NH4Cl and use the blue color to represent and use the green color to represent 14 NH4Cl. They had kept E. coli in 15 NH4Cl for a long period of time, more than 40 minutes. So 80 minutes. The total DNA was of 15 NH4Cl. Okay. Next, they give 20 minutes. Then these are transferred to 14 NH4Cl. What happened? for 20 minutes, 40 NH4Cl for 20 minutes. One generation completed. From 50 NH4Cl, the DNA, the initial DNA was like this, and there was mixture of this sort of DNA. From that DNA, these DNAs were formed. Okay. Again in NH4Cl, for 40 minutes, for extra 20 minutes, in 14 NH4Cl, for another 20 minutes in 40 NH4Cl, it was found that a mixture of these two were formed. This and heavy one and this. So initial is conserved and two new light and heavy DNA. So the parental is to new combination is 1:1. These were parental and this combination were formed. Okay. So what does it show? It shows that one strand of DNA remains fixed. To that strand of DNA, a portion of DNA replicates. And to the other strand of DNA, a portion replicates. So 4 replicated complementary strands form, then they get separated. 2 DNA were formed. Okay. This model is known as semi-conservative model. Semi-conservative model by Stahl and Messelson. This question came for a 3 mark in 2019 biology Cbse examination. I have explained the whole process. You might get what is the significance of 50 NH4Cl. You will write the answer like this. Nitrogen is a very important component of DNA. 15 NH4Cl being a heavier isotope, 15N being a heavier isotope of nitrogen was utilized so that the lighter and the heavier nitrogen of nitrogen can get incorporated into the DNA and can be easily centrifugated and separated using a cesium chloride gradient. Okay, like this you will frame the answer. So this proves the semi-conservative model. Next, a detailed description of how the replication takes place. You will get these two DNA. Now, since they are intertwined between them, these are two DNAs. Okay. What happens? First, the point of replication. The point of replication, where the replication begins, is known as OD. Origin of replication. For example, this point. From here the DNA replication will begin. This is known as OD, origin of replication. So the first phase is initiation. What are the events that takes place in initiation? Number A, the enzyme and the function. I will be discussing like this. The two strands unwind and separate. The two strands are formed like this, they separate. So what happens? From here they form like this. They form like this. They form a Y-shaped structure. They form a structure like this. This can be in more details, this can be drawn like this. And before this, these are the replication structure. Okay. This. In your examination, you need not draw this portion, I am just explaining. Like this, they unwind and open. What is the enzyme that is responsible for this? The enzyme is DNA guidase, DNA helicase. Okay. So the DNA guidase and DNA helicase unbinds or unwinds the two complementary strands of DNA. So as you know, the DNA have a strand named from 5 and 3 end. This remains as 5 and 3 end, and this is always complementary. This is 3 and 5 end. Next, number B. If you coil anything and then try to open it, they again recoil. Due to the intersurfacial tension, they again have a tendency to recoil. To prevent this recoiling, the enzyme SSBP is required. The two strands are prevented to recoil by tension, by surface tension. This is known as single stranded binding protein. SSBP. SSBP prevents the two coils to recoil, the two complementary strands to recoil. Next, this form a Y-shaped structure, and this is known as Y-shaped structure, and this is known as the replication fork. This model where these two are totally in a Y shape. This is Y, inverted Y. This is known as the replication fork. Next, I move on to the elongation phase. Initiation phase, okay. The two complementary strands of DNA are unwinded using DNA helicase and DNA guidase. The unwinded strands are kept together using by preventing the intersurfacial tension to act on them using the enzyme SSBP or single stranded binding protein. Now, the next phase is elongation. What do happen in elongation? The enzyme that acts is known as primase. The 3' free OH. What is the 3' free OH? This is a continuation of the previous one. So this OH is always free. Okay, as an ideal structure of base forms like this. See, here you have a free OH group at the 3' end. This is at the 3' end. This is present at the 3' end. This free OH group at the 3' end, this you need not draw. This is not in your syllabus. This is in the pharmacy syllabus. At the 3' end, the extra individual, the codons of the DNA, starts forming a replica. How? From the 3' to 5' end, suppose this was the A, T, C, G, it will form P, A, G, C. It will form a complementary strand. From 3' to 5' end, from the free 3' to the 5' end, this will form a continuous segment. Since this is complementary to 3' to 5' end, the 3' will form 5' and the 5' will form 3'. This will form a continuous formation from 5' to the 3' end. Okay, this is known as leading strand. The continuous secretion from the 3' to 5' end forming a new DNA strand with 5' to 3' end is known as leading strand. But from this 5' to 3' end, the strands that are formed are in fragments. For example, this totally unbounds to form to give a total synthesized complementary DNA, but they are forming fragments. A bit unbound, a bit of the this complementary DNA is formed. Again a bit unbound, this is formed. Again a bit unbound, this is formed. So it will number 3' to 5' end. It will be numbered, of course, because it is 5', it will form a complementary 3' end. These are formed up till the total, this is formed like this. They are formed using fragments. They are formed by fragments. Okay. These are the steps in elongation. From the 3' to 5' end, total synthesized DNA is formed with the numbering 5' to 3'. But from the 3' to 5' end, but from the 5' to 3' end, uneven or fragmented DNA is synthesized, that is numbered 5' to 3'. This is known as lagging strand. This is known as lagging strand. These fragments of DNA are termed as Okazaki fragments. Okazaki fragments. So, complete DNA never have DNA in form of fragments. They have a totally synthesized DNA. They needs to be total, continuous. So, to promote this continuity, the hormone that I'm extremely sorry. The enzyme that is responsible is known as DNA ligase. DNA ligase. What is the function? To join the Okazaki fragments to a continuous DNA. This is the function. So, after this, a total continuous DNA is formed. A total continuous strand is formed. So, this now gets separated and this will continue up to here. This will continue up to here, 3' and this from here, 5'. So, this then formed like this, a model like this. 3', 5' and the two DNA, 3', 5'. These are the two complementary DNA. From a single coiling, so what happens? Initially the DNA is coiled. From a single coiling, the binding gets separated and they unwind using DNA guidase and DNA helicase. They unwind, which is maintained using single cell SSBP. These are maintained by SSBP. Then from the 3' free OH, individual RNA polypeptide chain DNA polypeptide gets added to form a continuous synthesized DNA from the 3' to 5' end, and the newly DNA is from 5' to 3' end. This is known as leading strand. But from the 5' to 3' end and the DNA formed from 3' to 5' end, the DNA synthesis is fragmented. They are known as Okazaki fragments, and these fragments are then joined using DNA ligase. Okay, and two new DNA is formed. So today's video is up to here. Where in this, I have discussed about the DNA replication model, the experiment of Messelson and Stahl, and the replication fork diagram. Replication fork is extremely important for this thing. Since this is exam stamina 2020, I will be discussing the most important and the top rated topics for the examination of biology 2020 Cbse. So thank you and keep on studying. All the best for the upcoming examination of your biology. I hope you all are studying for all the rest topics along with biology. Keep on going and goodbye. Thank you.